Antibiotic susceptibility testing is too slow. Faster identification of microorganisms is now common, as many laboratories use MALDI-TOF or molecular technologies for quick and definitive identification of bacteria. Improvements in susceptibility testing have lagged, as we continue to use slow tests which have not significantly changed in decades. Rapid phenotypic testing can only be done on limited sample types using a dedicated platform that has not been widely adopted. Tests for rapid genotypic testing usually include only a few resistance genes and require confirmation by phenotypic testings. What are the prospects for fast susceptibility testing? Welcome to Editors in Conversation. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org and on Twitter at jclinmicro. I'm your co-host, JCM Editor-in-Chief Alex McAdam. This podcast is supported by the American Society for Microbiology, which publishes JCM. Please subscribe to the podcast and rate or review it. I'm joined by my co-host and a fellow editor of JCM, Dr. Ellie Thiel from the Mayo Clinic. Ellie, great to see you as always.
Good to see you, Alex.
We're also joined today by friend of the podcast and return guest, Dr. Trish Simner. Trish is an associate professor of pathology at Johns Hopkins Medicine, where she is also director of medical bacteriology and infectious disease sequencing. Trish, great to have you back.
Thanks so much for having me.
I'm also pleased to welcome first-time guest, Dr. Dan Rhodes. Dan is the section head of microbiology at the Cleveland Clinic, where he holds the Belinda Yen Lieberman, PhD, and James N. Lieberman, MD, endowed chair in clinical microbiology. Dan, thanks for joining us.
Thanks, Alex. Happy to be here.
Trish and Dan are the first and last authors on a paper in press at JCM. The title is Multi-Center Evaluation of the Acuitas AMR Gene Panel for Detection of an Extended Panel of Antimicrobial Resistance Genes Among Bacterial Isolates. We might need to limit the characters on the titles going forward. We'll get into the paper shortly, but I want to talk about speeding up AST more generally first. Before we do, we have a few disclosures to make. Trish is a consultant to Opgen, which manufactures the test we'll be talking about. And also, this study was sponsored by Opgen, so both Trish's and Dan's work was done with that support. So let's go ahead and jump right into it. And we'll start with you, Trish, and Dan, Ellie, feel free to jump in. Can you give us just a brief overview of the scope of the AST problem, the antimicrobial susceptibility testing problem, and antimicrobial resistance?
Well, I just got to say that's a loaded question, Alex. And if you get me on my soapbox, you might not get me off. So I will try to stay focused here. So if we just think about it, our conventional identification and antimicrobial susceptibility testing methods that we use in lab are our primary methods to detect antimicrobial resistance and is used to guide treatment of these infections. The problems with our conventional methods are that they're slow, and we'll dive into that a little bit further later. However, it is our tried and true method that physicians are experienced and reliant on. Luckily, over the last 10 years, we've observed the introduction of new tools that allow us to more rapidly detect antimicrobial resistance in the lab. These can include phenotypic or molecular methods. So rapid phenotypic methods can include methods like the PBP2A assay that detects the product of MecA. So if it's absent, you can predict that it's methicillin susceptible, whereas if it's present, you can predict methicillin resistance among staph species. Other phenotypic methods can include simple methods like chromogenic agars with antibiotics in them for selection, or the introduction of these novel phenotypic methods that have mostly been created for rapid AST from positive blood cultures, and these methods use novel methodologies to detect growth in the presence of antibiotics, so to detect that phenotype. On the flip side, we have molecular methods, and these can be performed directly from specimens or cultured isolates. In general, these molecular panels are focus on a few specific AMR genes or high priority AMR genes like MecA and MecC for methicillin-resistant Staph aureus, or VAN-A, VAN-B for vancomycin-resistant enterococci. From the gram-negative side, there's the predominant ESBL gene CTXM that can be on these panels, or the big 5 carbapentamases like KPC, VIM, IMP, OXA48, and NDM. What we're starting to see now is expansion of these panels to try to be more comprehensive from a molecular standpoint. Most of the panels to date that expand from the ones that I just discussed are RUO panels that detect additional beta-lactamase genes. In addition, we're starting to see whole genome sequencing becoming more commonplace in clinical microbiology labs. However, this method has mostly been implemented for outbreak or surveillance purposes. However, from that data generated, especially if you could do this more in real time, you can then query the genome for resistance genes to try to detect AMR, antimicrobial resistance, as well as for the potential to predict phenotypic AST results. However, with whole genome sequencing, the methods still are still too cumbersome at this point and require significant molecular and bioinformatics expertise. And there's other issues that we could delve into on that side for, how to predict the phenotype. But for now, I'm just going to leave it at that and just say that we've been lucky to see some new technologies come out that help us address the ability to more rapidly detect antimicrobial resistance than our traditional conventional ID and AST methods.
Thank you, Trish. That was terrific. Dan, give us an idea of sort of the problem that we're trying to solve. How long does conventional antibiotic susceptibility testing take? What's the kind of range, and what are the reasons why it is as slow as it is?
Yeah, typically it takes a couple days from specimen to susceptibility results, right? So first we inoculate the plate, then pretty much next day, typically 24 hours later, generally, we look at those plates, and if there's something that catches our eye as a probable pathogen, then we set it up for susceptibility testing. That takes essentially another day. So a full 24 hours, maybe as short as 16 hours to get results. So from an isolate to susceptibility results, usually it takes a day. As Trish mentioned, there's some tools we can use to try to find high yield markers of resistance, where we can infer resistance based on the presence or absence of certain targets. But conventional susceptibility testing, whether it's in broth or on an auger, typically takes about a day.
Thank you both. So we've got a good foundation here on the different alternatives to conventional testing. I turned to another expert, which is Twitter, and I posted a poll on Twitter asking which of these is most likely to replace current phenotypic methods for comprehensive, faster AST. And the choices were targeted genotypic testing like PCR, whole genome sequencing, and faster phenotypic methods. So before we talk about the results of the Twitter poll, why don't we start with you, Dan? Which of these do you think holds the greatest potential, and why?
I think your question was also for near term, if I remember right, because I saw that Twitter poll. And I think near term, I think lowest hanging fruit is faster phenotypic susceptibility testing. The one that came to market a few years ago and is still in the market is Accelerate. I know in Europe there's Q-linea, and there's other vendors that are trying to push into the space. For decades, people have been using rapid susceptibility testing on positive blood cultures by taking the blood broth, and instead of taking it to auger, taking it straight to a susceptibility method, whether that's like a Vitek 2 off-label or microscan or the old-fashioned disc diffusion. There's ways to do rapid AST, and I'm hopeful that in the future, as bacteriology becomes more automated, there'll be even better ways to expedite susceptibility testing. That said, I'm excited about tools like whole genome sequencing. I mean, one of the first times I remember running into Trish was at a ASM, maybe it's a general meeting or microbe. I can't remember what it was at that time, where she was talking about using a mini-on and detecting KPC within minutes from sequencing. So there's a lot of exciting opportunities, but I think for most labs, off-the-shelf solutions that have received FDA authorization are going to be most readily available soonest. And that, I would say, that would be rapid phenotypic testing.
Perfect. Thank you, Dan. How about you, Trish?
Well, Alex, I was torn at what to choose. Usually I look at those Twitter polls and I quickly respond, but I don't think there's kind of a one size fit all. Like this is a bigger problem. There are some organisms that are difficult to are unreliable in terms of standard AST methods to and correlating to clinical outcomes. A good example is Burkholderia cepacia complex, where our European colleagues UCAS doesn't even have breakpoints for this group because of the lack of reproducible methods and no convincing good clinical correlates with those results. And I know that CLSI is looking at that too. That being said, and so that's where I feel like, some of these molecular methods, especially whole genome sequencing, might be a tool that would be helpful to be able to provide more accurate information to help for clinical care, but we still don't have, outcome studies with these methods. And so I do think we need to look into that further. But in terms of, more direct response now, I think these rapid phenotypic methods are where our field is going. And CLSI and UCAS just came out with standardized disk diffusion methods from positive blood cultures. And I think in combination with total lab automation, you get this kind of affordable method, especially if you have total TLA and if you've invested into TLA, I should say. You might be able to, standardize those rapid disk diffusion methods in your lab a little bit easier. Or like Dan said, using liquid colonies or inoculums like from the sepsi typer, or, we're seeing other liquid colony methods like Qvella or the Accelerate Arc, that you take that cleaned up biomass from the positive blood culture and put it in your standard Phoenix Vitec, again, off-label after validation, of course. And then again, I also think, these targeted panels are also helpful in between. I think it also allows us to kind of target how we're reporting them if we know, and we also know exactly what we're looking for on those panels. So, that's helpful with whole genome sequencing. Although we think it can detect all mechanisms, it's not always the most ideal method for detection of, let's say, inducible resistance or efflux mediated resistance, right? So I think, we might think it's the broad method, but it's not detecting all methods, and it's only as good as our database as well. With a whole genome sequencing, detecting AMR it's helpful. There are some limitations there, but also the complexity of trying to predict the phenotype from that is not as straightforward. The first step is detecting whether you see the presence or absence of AMR genes. The next step is trying to predict and correlate to that phenotype, which we're just not there yet for certain drug-bug combinations.
All right, so do I hear a vote for rapid phenotypic in the near future with some very thoughtful comments about the potential for targeted amplification and for whole genome sequencing in the future, and for perhaps specific organisms in the near future? All right,
correct.
Good. Ellie, I know that you are the royalty of serology. Do you want to jump in on this? Do you have thoughts on which of these has potential?
Well, who am I to disagree with the experts, Alex? But I'm going to flip it on you. You posted the question, but what are your thoughts?
I think it's rapid phenotypic in the near term, and there's a lot going on here. Dan sent us a paper from our sibling journal, AAC, just before we started recording that looks very interesting on a rapid method for doing susceptibility testing by a phenotypic method from blood cultures. Carrie-Ann Burnham had a really nice paper in JCM where she showed that you could set up disk diffusion testing on colonies much younger than we usually set it up on, that you could use colonies as young as 6 hours old and still get accurate results from disk diffusion testing. So that lops potentially about a day off of phenotypic testing. So that said, and Trish, you were the editor on that, weren't you?
Not on that one,
Not
but
that one?
I did have a chance to review it thanks to your Twitter post on it.
Oh, good. Good. But that said, that's the near term. I think that multiplex PCR or other amplification methods and whole genome sequencing hold tremendous potential, and that there's a lot of development, a lot of energy there. So we may see that coming a little bit later. but I wouldn't be surprised if it really took center stage before too long. Twitter said, I was surprised how many votes we got on this. We got 379 votes, which I thought was terrific. 52% voted for faster phenotypic testing. 31% voted for targeted genotypic testing. 17% voted for whole genome sequencing. So pretty much in line with what we have been saying. All right, so let's get into your paper, if that's okay. Trish, why don't we start with you? Can you tell us what is the Acuitas AMR gene panel? How does it work? Give us an overview of the assay itself and a brief summary of the workflow.
Sure. So the Acuitas gene panel falls into one of our targeted panel molecular methods. So it's a multiplex real-time PCR assay for the detection of up to 28 antimicrobial resistance markers. It's in a 96-well plate PCR format. It can be performed on isolated colonies from either blood or MacConkey agar for select Enterobacterales, Pseudomonas aeruginosa, and Enterococcus faecalis. So that's not, what's unique here is, this expanded AMR panel. But what is extra unique about this panel is that when the company went to the FDA, they also wanted to try to associate detection of these AMR markers with not susceptible results with the antimicrobial agents based off of the drug-bug combination or the drug-bug combination detected. So they can then take those AMR markers and associate that with not susceptible results for up to 9 antimicrobial classes / subclasses. So this assay is performed on the QIAGEN Advanced Excel, where you do your nucleic acid extraction from colonies, from pure culture colony growth. And then from the extract, you'll then use the Applied Biosystems QuantStudio 5 instrument for the real-time PCR instrument. So this method really is a semi-automated approach where it does require interventions of the technologists. So what you're going to do is you're going to take your isolates, your pure colony, or you're going to pick up to 3 to 5 isolated colonies of your organism of interest. You're going to do the extraction component. You'll take your extract from your isolate, and you're going to mix that with the master mix that's part of the kit. And then you're going to pipette that mixture into 3 columns of the 96-well plate. So for each plate, you can test up to 4 isolates. And in that panel, they have proprietary dried primers and probes. Once you've inoculated your plate, you're going to seal that, centrifuge it, and then place it on the QuantStudio instrument for the pre-programmed real-time PCR assay on the instrument. Once the run is complete, the results can be exported to Excel, and then you're going to open the Acuitas AMR gene analysis software that will then generate a report based off of the results. And so on that, in the system, you're going to use a drop-down menu to be able to pick the organism. So you need an upfront identification method, whether that be a rapid MALDI or a molecular method for the ID. And then based off of the assay, it'll correlate the results as either detected or not detected as appropriate for the antimicrobial resistance gene and the bug. And then it'll also list the on-panel genes and associated not susceptible results for the specific organism selected as appropriate for the clearance of the assay. So the whole process from your isolate to result takes about 2.5 hours with approximately 30 minutes of hands-on time.
So, Trish, can you kind of break it down for me a little bit? What is different about this molecular PCR-based AMR gene detection assay that allows the company to say, susceptible, not susceptible? How is that different than other PCR assays that are detecting AMR genes?
That's a great question, Ellie. So I think most of the PCR assays on the market that detect an AMR gene have been FDA cleared to be able to say whether it's present or absent, detected or not detected, is where they stopped at. Whereas for this assay, they went on to be able to correlate the detection of that gene with a phenotypic resistance result. Now, I want to be clear here, this is not a prediction tool. So the presence does not predict resistance, the absence does not predict susceptibility. I know that's complex. So what they went for was an association. So a positive predictive value of at least 80%, that if that resistance gene is detected, there's at least an 80% likelihood that that isolate is resistant to that particular agent that it's predicting, or associating, I should say, not predicting. So it's an association rather than a prediction, and it's only associating with the presence for predicting not susceptible or resistant results. So to your point, Ellie, is that most molecular tools so far might be used in that manner in the lab, that the presence, predicts resistance, the absence may or may not predict susceptibility, depending if you're talking about a gram-positive or a gram-negative. That's how some of these methods are used clinically, but they have not gone through clearance by the FDA for that purpose. They're really just gone through whether they compare to a PCR or another method that detects the presence or absence of that gene.
Got it. Thank you.
It's a subtle distinction. And I think I'm going to ask Dan in a second to tell us about the study design, but when you do, Dan, talk a little bit about the reference method, the gold standard that was used that helped them to be able to make that claim. But you don't have to start there, but just give us a little overview of the study design.
Yeah. So the study included 4 sites. Trish's site was one of them. My site was one of the sites. 4 sites in total, and they used both retrospective isolated colonies that they had collected that we knew had some markers of resistance or had some phenotypic evidence of resistance, and also prospectively collected isolates. So there were, I had to take notes, there were 584 retrospective isolates and 83 prospective isolates. Now, some of those were tested with replicates. So in total, there were about 1,400 tests performed on those 600 or 700 isolates. We, as Trish was describing, you can test up to 8 isolates on those 96-well trays at a time, 4 isolates per tray. So this is really a small batched workflow. And as Trish mentioned, it takes 2 to 3 hours from start to finish. So the automated extraction was performed. That took an hour, and then the scientists had to move it over to the thermocycler, which takes about another hour. And then data analysis is also another transfer step. That's the last step to make the interpretation based on the genes that are detected or absent. So the comparator methods were mostly standard of care for susceptibility testing. So Microscan, which is a commercially available antimicrobial susceptibility test system that's FDA authorized, was used for the gram negatives, for example. MALDI was used for identification, which has become standard of care over the last decade. And then whole genome sequencing was also used, which isn't really standard of care, but it was the reference method for detection of the resistance markers, and also for identification of the isolates.
Got it. So I just want to make sure that we're clear. The phenotypic susceptibility testing was the reference method for susceptibility testing.
Right.
The whole genome sequencing was the reference method for the presence or absence of the AMR gene. Is that
Correct.
right?
Yes.
Okay.
Yes.
Got it. Anything to add there, Trish?
No, that was perfect.
All right, great. All right, so that's the method. That's the study design. What did you find, Trish?
Well, so the panel went to the FDA with 31 AMR markers. In the end, 28 AMR markers were cleared by the FDA on this panel, where they achieved at least 94.4% positive percent agreement and 96.5% negative percent agreement.
Can I jump in?
Yeah.
Where'd that 94 point something come from? It's
So
in the paper, but
yeah. So there was some small nuances where the FDA had approved, I think it's MCR1, would need to verify it, but it fell just below the 95% PPA cutoff, and they agreed to allow clearance of that marker despite it falling slightly lower as an overall. For the individual organisms, for the great majority of them, they were above that 95%.
thank you. Sorry to break in. Please
No,
go ahead. Yeah.
it was an interesting thing to explain, as an author too of the paper itself. So, and then, in thinking about, remember I said there was 31 that went to the FDA. The 3 that did not make the FDA clearance were GES, SPM, and MCR2, where GES and SPM did not meet the PPA requirements for acceptance, and then MCR2 was not included because they did not have enough clinical isolates of MCR2 included in the study, and so that's the reason why those 3 markers are not going to be on the FDA cleared version of this assay. So as I mentioned, just detecting the genes was the first step, but they also wanted to not only just detect the genes, but then correlate them to not susceptible results. And so based off of the additional requirement of correlating to not susceptible results, up to 18 AMR markers can be reported per species. So the correlation with phenotypic AST made it so that some markers are cleared for some bugs, whereas not for others. So just to make it clear that they required both of those criteria, so the PPA and NPA for detection / absence of the genes, and then also requiring at least 80% positive predictive value to predict not susceptibility correlating to the phenotype to be approved for use on this assay. So it required those 2 requirements. And so because of that, at a minimum, a single antimicrobial agent can be detected and associated with not susceptible results from the acuitas panel, such as vancomycin with the detection of VAN-A among Enterococcus faecalis, whereas a larger set of antimicrobial agents can be associated with not susceptible results among common Enterobacterales like E. coli or Klebsiella pneumoniae, as well as with Pseudomonas aeruginosa. And the associated agents for those organisms include some of the most commonly prescribed antimicrobial agents, including beta-lactams, fluoroquinolones, trimethoprim sulfamethoxazole, and the aminoglycosides. So I do want to point out here that this is an adjunct test. And so even though you are able to kind of associate a phenotype with the detection of these genes, that you still need to follow up. It is not a replacement to traditional phenotypic AST. So you would still need to take your isolated colonies and perform your traditional phenotypic AST methods in the end when using this assay. But overall, it looked like a pretty accurate method to detect resistance genes and also correlate with an associated not susceptible result when detected.
Thank you. And Ellie and Trish know that I rarely like the way that discrepant results are handled in papers. Dan, I don't know if you've heard me preach that sermon, but in fact, I liked the way that discrepant samples were handled in this study. So Dan, since I was too lazy to do it, tell people what a discrepant result is and how you handled them in the paper.
Yeah, so discrepant results are things that don't really make sense when you look closely or when you compare to the reference method. It was essentially wrong. Your test was wrong or deemed to be wrong. So the whole genome sequencing was helpful in finding these discrepant results in this study. There were about 50 discrepant results identified with the over 1,000 tests that were performed. And some of them looked like they were sample mix-ups, which always make me cringe because I like to think that we don't mix anything up in the lab. So that was concerning. There were 9 identification errors where something happened between the MALDI identification and then the whole genome sequencing. So it looked like there was a specimen mix-up at some location along the way. And then there were also some inconsistent results with sequencing, where a gene was detected one time and then retesting with sequencing, the gene wasn't able to be detected. So there were those errors identified. There's also, some things I thought were interesting. I want to point out that we made it about 20 minutes in and nobody said COVID yet. So here is my reference to COVID. So one.
We can edit this out later.
Exactly. So, one example, there was kind of baseline fluorescent drift in the PCR that the software interpreted as positive, and that wasn't the case. And, this takes me back to like flashbacks of last winter with TACPath and the challenges with false positivity due to baseline drift. But yeah, now that that's out of the way, there was also some oligonucleotides that were in the OpGen assay that didn't match with the target. So the genes were present, but the variant in the genome didn't allow annealing of the primers of the probes. So it wasn't detected. That was able to be discerned by looking at the whole genome sequence. And interestingly, one of the replicates that was actually replicated 7 times had a CTXM gene that was identified by OpGen, but it was actually a slightly different variant of CTXM than OpGen thought it was. So those are some of the examples of the discrepancies that were identified.
Thank you. And then, having analyzed those discrepancies, did you go back and recalculate your positive and negative predictive value, which are analogous to sensitivity and specificity?
Trish, you want to take that one?
Yeah, I can totally jump in. Yeah, no. So to Alex's, happiness here, we did not do any discordant analysis and have to recalculate PPA and NPA. We actually just are based off of, OpGen and discussions with the FDA for the paper and for the clearance of the assay, they decided not to pursue discordant analysis and thought that the discordant analysis wouldn't have impacted the results that much and decided not to pursue it. So really the results being presented are the initial attempt at running this assay with those results and understanding that, yes, there might have been some sample mix-up or some PCR drifts occurring, but that the data is what it is, and so no discordant analysis was pursued there.
Oh, I'm so happy. Thank you for picking a reference standard that you trusted and sticking with it. We've touched on workflow and reporting. Anything to add about workflow and particularly about reporting, Trish?
Yeah, I think, what this paper shows is that this is an accurate method that we can use to detect these AMR genes among the specific organisms and can use those data to associate with not susceptible results. But how do we implement this in the lab, with the main goal of coming back to our question of trying to be more rapid than our conventional ID and AST to impact care? This is, as we mentioned, a semi-automated method that requires intervention. It also requires batch testing because at a minimum, if you run one 96-well plate, you need to fill it up with at least 4 isolates to make it cost effective. I mean, you could run one, but then it's wasteful, right? So how do we envision utilizing this, whether it be, whether it be the sample type or the patient population that we focus the utility of this test on. And then from a lab workflow kind of question, how do we do that? And then from a clinical utility standpoint, this also needs to be evaluated. How does these results impact treatment? How does it impact stewardship and infection control initiatives? The nice thing about this panel is that it can detect a wide array of AMR genes as well as some, emerging resistance genes like MCR1. So this could be helpful for infection control. The other thing is from a clinical utility and getting back to our question about phenotype versus genotype earlier, the phenotype is nice because it's a broad catch-all, like it'll detect whether it's susceptible or resistant to the 3rd generation cephalosporins. But what we're starting to see is that the mechanism does matter. And, we've seen new IDSA treatment guidelines that come out that have different recommendations for third generation cephalosporin resistance mediated by ESBL versus, the same resistance mediated by AMP-C. So I do think that there might be some information in here over and above the phenotype that can be helpful for treatment decisions that need to be looked at further. And then the last part of that is the reporting aspect that Alex alluded to. I think we're starting to see these AMR panels broaden in the lab. And I think, and some of these panels, like the blood panels, people understand the clinicians understand the organisms, but sometimes aren't as familiar with the antimicrobial resistance genes detected. And it can be a little bit of an alphabet soup, right? Me studying AMR genes, every once in a while I come across one like, what the heck is the three gene acronym, what is it? So I think that we need to be more critical and provide more assessment or thought into how we report these AMR genes to patient-facing clinicians so that it can be used appropriately for clinical care. And as part of the Antibiotic Resistance Leadership Group Diagnostic Committee, we actually did a survey recently on this and are going to provide some consensus statements to hope that we're not just listing these, you could imagine 28 AMR genes detected, not detected. How does that impact care, right? So we want to provide valuable information in reporting these results. I don't think we know what's the best way, especially for a broad panel like the Acuitas panel yet. But I think if you are considering a lab considering bringing this in, you need to work closely with your stewardship committee to devise reporting strategies that are consistent with your formulary, because in here you're going to be reporting associated not susceptible results for antimicrobial agents. So make sure that it's consistent with your formulary, as well as make sure it's consistent with antibiotic treatment guidelines at your facility, and work closely with the stewardship team on, the best way of reporting these results to benefit patient care.
Thank you.
So
yeah,
maybe I'll jump in and maybe I'll direct this to Dan. Looking at the panel, it's a broad panel, so that's good. But are there any targets you would have added or maybe not removed? We probably don't want to remove targets, but there's no Van B, for example. There's no Staph aureus, MecA, MecC targets. I found that curious, but just wondering what your thoughts are on the composition of this particular panel.
Yeah, that's a great question, Ellie. And I think if you take a step back, I think the reason is probably the goal is to get this through the FDA, right? At the end of the day. And I think that drives a lot of decisions that are made for a lot of our in vitro diagnostic assays. We've talked about how this is one of the first to be able to correlate marker of resistance with actual phenotype. I do that all day long, in real life. If we find KPC, we're going to say, expect the carbapenems to be resistant. Somebody's doing that interpretation. But in this case, the vendor, the manufacturer is actually taking it on themselves to take it to the FDA and say, can we say this out loud? So they also have to decide, what's the use case or what's the targeted use case for this. And I think if you look at the panel, it's targeting probably mostly urine isolates, right? With Enterococcus faecalis, Enterococcus faecium wasn't included. And faecalis is a common urinary tract infection cause, whereas faecium is more uncommon. And with Van B, it's definitely causes vancomycin resistance, but we don't encounter it quite as commonly as Van A. So I think it's just at some point you have to draw a line and say, we're going to go after this, and we're going to let this go for round two, for the next version of the assay or the next iteration. So yeah, there's always more that we'd love to include, when we're looking at tests. But until we get to whole genome, which includes, whole plasmid sequencing, there's always going to be gaps.
Got it. Makes sense.
Can I add to the wish list, Ellie?
Yes.
Okay. So one of my things that I would love to see on these panels, and I realize it's a challenge to our industry colleagues, is inclusion of other ESBL genes like TEM and CHEV variants that are ESBLs. I realize there's a small difference between it being a narrow versus an extended spectrum beta-lactamase among TEM and CHEV variants, but although CTXM is predominant, I do think we are seeing TEM and CHEV ESBL variants. And if we think about it in terms of treatment guidelines, especially positive blood cultures, if you're seeing an ESBL, you want to, the recommendations are meropenem. So right now we're limited to CTXM only, and I would love to see the inclusion of other ESBL variants like TEM and CHEV, as well as plasma-mediated AMP-C genes, which on this panel there is a few of them like CMY and DHA. So I'm excited to see the expansion of the panels in that direction, but some of the genes that are on my wish list.
Sounds like there might be a panel 2 in the future.
All right. Well, when there is, we'll have you back to talk about it. I think we've done a great job covering this, and this is probably a reasonable place to stop. Ellie, always a pleasure.
Same here. Great to see you guys.
Trish, thanks for joining us again.
Thanks for having me back. That means I didn't fail the first time or the last couple times. So I'm always encouraged when you ask me back. So thank you for having me.
Dan, thanks for joining us. We'll have to have you back another time as well.
Thanks so much.
And thank you for listening.